Okay, a couple of announcements. One is go to your sections today and or tomorrow. If you don't go to your discussion sections, and make sure you're there and make sure your presence is known, you'll be dropped from the class. It's the only way we have of keeping track of everybody. So be sure you go through to your discussion sections. And labs start this week. There's a lab lecture tonight, and tomorrow through Friday there are labs. So don't forget to go to labs, all right? I will have office hours today from 9 to 10 and also tomorrow from 9 to 10. It takes me a little while to walk down from here to 2084, so don't give up hope. And if the door's open, go on in and get a seat. There were a lot of people there on Friday and maybe not enough seats. We can try to deal with that. It's difficult. But the Tuesday office hours are usually very scantily populated. How does that sound? There are not a lot of people come on Tuesdays. So if you can come and you have questions, it's a good day to come to lab, to office hours. I was looking at the webcast of my Friday lecture, and there were a couple of things I said that weren't quite, one was not quite accurate. It was true, but not quite accurate. And then I wanted to just make another comment on another one. When I was talking about hemoglobin, I pointed out, and the structure of the not the structure, the figure of the molecule is shown in the handout. And next to that, there is a group which I've said was heme. And I said that the heme is important because the oxygen binds to the heme. That's true, but what the oxygen is really binding to is an iron atom. If you look closely at the structure of the heme molecule, there are four. It's a planar molecule with an iron in the middle. That iron is what directly binds the oxygen in hemoglobin. And we'll talk about heme groups later on as well. The other thing I wanted to mention, which I did not mention, when I talked about the Anfinsen experiment, the Anfinsen experiment was the experiment where he unfolded this protein, this enzyme, it lost activity, and then it refolded, and the activity came back. And I said when you unfold proteins, this is called denaturation. There are other ways you can denature proteins. Most denaturations are irreversible. The classic example of denaturing a protein is when you take an egg and you throw it in a frying pan. The egg white turns, goes from clear to this whitish color. You're denaturing all the proteins in the egg. And heating proteins is a very common way of denaturing them. Other methods involve treating them with organic solvents. You'll do some of this when you do the photosynthesis experiment, and often taking the pH down to pH 1 will also denature proteins. These processes are irreversible unless somebody can take an egg, fried egg, and make it back into a real egg, okay? That's a good trick. So I want you to remember that the conditions that were used in the Anfinsen experiment were, quote, mild, and that protein could be renatured, but generally speaking, it's not done, can't be done. Okay, so today I'm going to talk briefly about carbohydrates, even more briefly about nucleic acids, and then go on to start talking about cell structure and function of components within cells. Carbohydrates. Everybody knows what carbohydrates are. They are affectionately known as sugars. And carbohydrates have a general formula, CH2O. They come in multiples of CH2O. You can have 3 of those, in which case, if you have your handout with you, you will end up with a triose sugar. You can have 4 carbons, which is a tetrose sugar. You can have 5 carbons, which is a pentose sugar. 6 carbons, a hexose sugar. Already, there's a question. Yes?
Shouldn't the fructose be a hexose, not be a pentose?
Fructose, that's right. That's a mistake. Fructose is a hexose, just the way glucose is a hexose, because it's got 6 carbons, which I will say in a minute. Thank you very much. Hexose. So you can have sugars with 3, 4, 5, 6, even 7 carbons. As shown in figure 25, there's an example of a 3-carbon sugar, glyceraldehyde, a 5-carbon sugar, and a 6-carbon sugar. All sugars have either an aldehyde group, in which case they are aldoses, aldose sugar, or ketone groups, in which case they are ketose sugars. You can see from these structures that I put on the board why these molecules are very hydrophilic. They've got a lot of OH groups on them, 4, 5, 6 OH groups. So you expect these molecules to be very soluble in water. Fructose and glucose have the same chemical formula, same structural formula, C6H12O6. But they are very different molecules because of one having an active ketone group and the other having an active aldehyde group. There should be 1, 2, 3, 4, 5. There should be another one. Okay? You have the structures in the handout. The handout is right, and I am not right. Okay? There's another carbon. Figure 27 shows that you can have isomers. Carbohydrates come in isomeric forms. For example, glucose, galactose, and mannose are all very similar, except the position of an OH group changes. So they are isomers of each other. And what's shown in that figure as well is that these sugars in solution form rings. The ring formation is shown in figure 26. This is not a big point, but it's a point you should be aware of, and you probably have seen this elsewhere. So ring structures will have groups sticking above the ring and sticking below the ring. In figure 26 for glucose, when glucose forms a ring structure, there's an OH that can be above the ring, in which case it is beta glucose, and there's another form of glucose where the OH is below the ring, and that becomes alpha glucose. Now, that one little change, say between, if you compare glucose and galactose, it looks very minor, but in biology it's major because for example, an enzyme that will react with glucose may not react with galactose because of the different stereochemistry of the galactose molecule. Now, you're probably familiar with glucose and fructose. What I know you are also familiar with are disaccharides. Disaccharides are shown in figure 28. And let me say there are lots and lots of different sugar molecules. We will really be focusing on glucose. And much of the biology, that is the metabolism that occurs in cells, involves glucose, but be aware that there are lots and lots of other sugars. Sucrose is a disaccharide formed from glucose and fructose, maltose, glucose and glucose, and you will be studying maltose indirectly in the lab when you study the enzyme amylase, which breaks down polysaccharides. Lactose, which is milk sugar, glucose, and a galactose. The coupling, the bonding between these two monosaccharides is a glycosidic bond. So when you build a carbohydrate polymer, you are building it from glycosidic bonds with one monosaccharide and another monosaccharide forming a covalent bond, water being split out just the way I talked about in the first lecture. Also, you have to be aware of the kind of glycosidic bonds that can be formed because you've got a lot of OHs. So, for example, sucrose is a 1,2 glycosidic bond. This is less important for you to know. Okay, now we come to the polymers. The polymers are shown in figure 29. There are really 4 carbohydrate polymers that we'll consider. The first one is amylose, which is a glucose polymer. Fortunately for you, all the polymers we talk about are glucose polymers. This does not mean there are not other kind of polymers in biological systems. For example, there are fructose polymers, but we don't talk about those. You can see that amylose is shown in figure 29, and it is a linear polymer. It actually forms some sort of helical structure in solution, but it is a non-branched polymer. Contrast amylose with amylopectin, its sort of brother. That is a molecule that has branching. So in amylose, you have 1,4 linkages to form your polymer, whereas in amylopectin, you have 1,4 and 1,6, and the 1,6 are the branches. Amylose and amylopectin are found in plants, and when one says starch, plants make starch in the light. They synthesize starch in the light. They're synthesizing amylose and amylopectin, and those of you who think about it might be aware that plants don't go to sleep at night. They continue to metabolize, and what they're doing is during the day they make starch in their chloroplast. They make amylose and amylopectin. At night, they break down the starch to glucose residues, which are metabolized by reactions that are common with animal cells, and we'll talk about that later. So amylose and amylopectin, branched, unbranched, but alpha-glucose polymers. We make a storage polysaccharide as well, and that's glycogen, which is shown in the upper part of figure 29. Glycogen is also an alpha-glucose polymer, but it is said to be highly branched. There's more branching in glycogen than in amylopectin. We make glycogen also for storage of carbohydrate molecules, I think primarily in our liver. And when we need energy, because that's what you get when you break down these compounds, you can break down your glycogen, your stored glycogen. Because these molecules are broken down by enzymes and the glucose units are further metabolized, we call them energy storage polysaccharides. There is one polysaccharide which is not an energy storage polysaccharide. It is a structural polysaccharide. It is the most prevalent organic molecule on the face of the earth, and that is cellulose, which is found in all higher plants, trees, for example. Everybody knows where cellulose is in trees. Cellulose is a glucose polymer, but it contains beta-glucose. So remember, I pointed out you can have alpha-glucose and you can have beta-glucose. Alpha-glucose is the most prominent compound in say, us, but plants use the other form of glucose, beta-glucose, to form a 1,4 non-branched polymer. Cellulose molecules are actually held together by hydrogen bonds, and they are found in, cellulose is found in plant cell walls, which is very important for the structural integrity of plants. Interestingly, an enzyme like amylase, which you're going to use in lab, which will break down alpha-glucose polymers, will not break down beta-glucose polymers. In fact, it is very difficult to break down cellulose. So this is an important table because it summarizes everything I've said about the polysaccharide polymers, and you should pay some attention to that, okay? That's all I can say. Okay, that's all I'm going to say about carbohydrates. We will talk about carbohydrates indirectly when we talk about metabolism. Nucleic acids. Next, the building block of the nucleic acid is the nucleotide, and that's shown in figure 31. What is a nucleotide? It's a sugar. There's a sugar residue, a 5-carbon sugar, a pentose sugar. There's a base. We'll talk about what those are. And then there's a phosphate group. The first thing I want to point out, which is also pointed out in figure 31, is this phosphate is a phosphoester. It's a bond between a phosphate group and an OH group on the sugar. And phosphoesters I talked about last lecture in lipids, and I said you're going to be seeing these again. And there it is, phosphoester bond. The bases. I don't expect you to memorize the structure of any of the compounds I'm talking about today. You should be aware of the difference between DNA and RNA, and basically I've put that on the board for you. It's also given in figure 31C. The bases and the two sugars are shown in figure 31, the middle of that page, where you have deoxyribose in DNA and ribose in RNA. The difference between those two is there's an OH in one and an H in the other. And then there are 5 different bases. Some are more complicated than others. The purine bases are more complicated than the pyrimidine bases, and they go by their letter names, A, G, C, T, and U. Adenine, guanine, thymine, uracil, and cytosine. DNA contains AGCT. RNA contains AGCU. The sugars are different, deoxyribose and ribose. That's the fundamental difference between DNA and RNA. Everybody who's a biology student at Berkeley has to know the difference between DNA and RNA. Otherwise, we're not doing our job teaching you, okay? The interesting thing about the DNA molecule, and this was discovered in the 1940s by, I think, a Columbia biochemist named Chargraf, was that in DNA, the amount of A, there's supposed to be an A there, always equals the amount of T. And the amount of G always equals the amount of C. And that's in DNA from all kinds of different organisms, bacteria, plants, animals. That rule holds. That's Chargraf's rule. The reason this is important is when you want to consider the structure of these molecules, you have to come up with a structure where the amount of A equals the amount of T and the amount of G equals the amount of C. Okay, so we'll have to do that. Not us, but the guys that were doing it. The way you get a polymer from these nucleotides is you string them together, both in RNA and DNA, by phosphodiester bonds. Again, something I referred to in the last lecture. And that's shown in figure 31B. You have a phosphate group on one sugar, on one nucleotide, a phosphate group on a second nucleotide. They form a phosphodiester bond. They split out water. So that is the way you polymerize nucleotides to get DNA and or RNA. How many people have heard of Rosalind Franklin? Most people have these days. Rosalind Franklin is shown in figure 32A. Rosalind Franklin worked on the X-ray structure of DNA. I am sure most people know this story. In fact, she had, and what's shown in figure 32A is one of those diffraction patterns that I mentioned last time for the protein crystallography. She had a diffraction pattern for a relatively pure crystalline form of DNA. Her data, and I could be slandered for saying things like this, but it's true. Her data was stolen by Jim Watson and used by Watson and Crick to propose the double helix structure for DNA, which is shown in figure 33. It's shown in figure 34. This is a structure where you have two helices wound around each other. One, I'm not going into any much detail because you're going to hear much more in the second series of lectures. One helix is running in that direction, one helix is running in that direction, and what's holding these two helices together are hydrogen bonds between A hydrogen bonds with T and G hydrogen bonds with C, and this explains why there are always equal amounts of AT and versus GC in DNA. Of course, this structure has become probably one of the greatest findings of certainly of modern biology and modern science in general. It has spurred all kinds of activity, ultimately leading to the Human Genome Project, where all of the information in these DNA molecules has been elucidated. So it really is just a revolution. 1954. If you ever want to look at the paper by Watson and Crick, it's a very short paper. In nature, it's like 2 pages or even 1 page. And this is, as I said, it's revolutionary. And it's amazing that they could, in 1 or 2 pages, put the structure out and propose ideas about how DNA gets replicated, which is very important. Figure 35 shows you that this thing, the nucleotide, it appears often in biology. For example, a molecule ATP, which we have talked about, which we will talk about extensively, is based on a nucleotide. It's called a nucleotide triphosphate. There are 3 phosphate groups in ATP. You'll hear more about that. Another molecule, acetyl coenzyme A, or coenzyme A, has again the same base. It's got the adenine, the sugar, phosphate, and then other things. And this is a very important molecule in metabolism, intermediary metabolism. And then there is a cyclic form of the compound AMP, which is shown at the bottom of figure 35, which you will hear about, I believe, when you hear some physiology. This is a sort of a signal molecule in cells. Okay, this is the end of molecules. And when I finish a section like this, if you have your canary book with you, I'll call it the canary book, I have at the end of each of these sections a series of review questions that you're supposed to just do. Maybe you can talk about them in discussion. They're for things that I think I have covered and that you should be aware of. They're very straightforward, but they should make you focus on the material and help you review that material. Now what we're going to turn to is the cell structure. And I'll just very briefly give the 4 basic principles on cell theory that have sort of also drive most of modern biology. Very simple principles that I'm sure everybody is aware of, but here they are. Cell theory, okay. Number 1, all living organisms contain at least 1 cell. Trivial, right? Okay. Number 2, chemical reactions of living organisms take place in cells. And we will talk about some of the chemical reactions, metabolism, that are occurring in cells, primarily reactions that are producing energy for cells. Number 3, cells arise from other cells. Well, that was not believed early on. There was a sort of a spontaneous generation of cells. Louis Pasteur proved for once and all that cells arise from other cells. And finally, 4, cells contain information, and that information we can now say parenthetically is in DNA molecules. That is passed from one generation to the next. Obviously, this is heredity, and this is the main topic of this middle of the class. All cells share 2, and I can even make it 3 essential features. Number 1 is all cells have an outer membrane known as the cell membrane or the plasma membrane, and this separates the outside world from the inside world of a cell. So what we have here is a cell membrane surrounding, this is a cell. And this is the in, and this is the out. I would say initially, right now, that the cell membrane has to allow things to cross. It's a barrier so that some things can't come in, but it is absolutely essential that all cells have ways of moving molecules across this membrane, and A to A inside, B to B inside, or vice versa, C to C outside. And we will talk later, at the end of the week, when I talk about membranes, on some of the function of this and other membranes. So we've got an outer membrane, a plasma membrane. The second feature that all cells have in common is they all contain genetic information. that directs all of the cell's activities and allows the cell to reproduce. Not a surprise. And I could mention one other thing, but maybe it's trivial as well. I like to say, I like you to think that all cells have ribosomes. Ribosomes are particles that are required for the synthesis of proteins, but most people wouldn't discuss that. But we're going to spend a lot of time talking about ribosomes today and for a while, so you're going to hear a lot about ribosomes again in the second part of the class. When one looks at cells, particularly the organization of the genetic material that is in the cell, one can characterize cells two different ways. There are prokaryotic cells, in which the DNA, and I'm talking about DNA, I could say genetic material, but I'm talking about DNA. The DNA is organized into a large single molecule. There's one single molecule of DNA. We can call that a chromosome. If you would like to call it a chromosome, that's all right. And that DNA is circular, one piece. And that's just in the cell. There is no specific membrane around a prokaryotic. So here's a prokaryote, and here's its DNA, okay, in the cell. Then there are cells that are called eukaryotes, or eukaryotic. Consistent. Eukaryotic cells, where it gets a little more complicated. Most of the DNA in a eukaryote is linear. And I emphasize the word most, and I will come back later and tell you why I don't say all. I say most of the DNA is linear and forms a number of distinct chromosomes. How many chromosomes do we have in our cells? 46, right? 2 pairs of 23. So we really have a cell, and it now has 46 of these things, okay? Not just a single circular piece. In both cases, these DNAs are complex with proteins. There are specific proteins that interact with DNA and play an important role in regulating its activity. But more importantly, the DNA in a eukaryotic organism is surrounded by a membrane system and is enclosed in what is known as a nucleus. So we have a major difference between how the DNA is organized in a eukaryote and where it is localized in the cell. There is no membrane around the DNA in a prokaryotic organism. There's another way that people often characterize cells. It's not based on the structure at all. It's based on their metabolism. And I can put that up now, or I can put it up later, but it's probably appropriate since we're talking about how one names different kinds of cells. We have cells which are known as autotrophic. And autotrophic cells are self-feeders. That's what this means, self-feeders. What do self-feeders mean? It means these organisms, these cells, can synthesize everything they have to synthesize within them, from simple compounds like CO2 and things like that, and other simple organic compounds. They're not using complex organic molecules for the synthesis of other things. And there are two kinds of autotrophic cells. There are photoautotrophs, which in addition can use sunlight. Well, you can guess what those guys are, right? Plants, algae, things like that. And there are chemoautotrophs. which are using, which are carrying out, again, a synthesis of complex molecules from CO2, but using inorganic compounds in much of this, use inorganic compounds in their synthesis. And we won't talk much about chemoautotrophs. We will talk a lot about autotrophs. Then there's a second group of organisms which are known as heterotrophic organisms. And these are organisms which use compounds for growth that have been produced by other organisms. They're not self-feeders. We are heterotrophs. We cannot make proteins, nucleic acids, lipids from CO2. We have to take in more complex molecules, break them down, and use those to make our biomolecules, complex, I'll say, relative to CO2. The only reason I put this up is one encounters in the literature often the terms heterotrophic or autotrophic, so it's a good idea for you to be aware of them. As you will see, and you can look at figure 36, table 36, and it gives you a summary of properties of prokaryotes and eukaryotes. And prokaryotes are much simpler. They do not have a membrane-bound nucleus. This is their most distinguishing feature. They are a lot of other things they don't have. They're much smaller than eukaryotic cells. Their method of division is different than a eukaryotic cell. If you compare figure 37, an animal cell, a plant cell, and a bacterial cell, bacteria are prokaryotes. The complexity of a prokaryote compares, it's worlds different than the complexity of a plant cell or an animal cell. What you see in both the plant and animal cell is an internal structure that is very complicated. Not only is there a nucleus, there are other things, other membrane-bound components in the eukaryotic cell, all of which are absent from the prokaryotic cell. Okay, how do we study cells? Cells are complicated. When you look at something like a plant or an animal cell, you see a very complicated structure. There's the old way and there's the newer way. In the old days, biologists, once they got a microscope from Leeuwenhoek in what, 15-something or something, you could look at cells. And you could look at cells, and when you did this, you saw a lot of, people started to see a lot of this internal structure and realized that eukaryotic cells were very complicated. But there are methods from using microscopy for identifying some of the components in a eukaryotic cell. You're going to do that in lab. You're going to do an experiment on mitosis where you stain the DNA in the nucleus and you watch it divide. Okay, so there are specific stains, compounds that react with specific things within the cells which allow you to identify them. Another intrinsic, that is something that where you don't have to even add anything, is if you look at plant cells and you want to look for a chloroplast, well, all the chlorophyll in a plant is localized in the chloroplast, and under certain conditions you can get that chlorophyll to fluoresce, so you will get light out from that, which you can detect, and very nicely you can say that's the chloroplast in the plant cell. The more modern method of studying cells is a biochemical approach, something that I have spent my life doing, which is smash and bash, you can call it, where you take your biological material and you break it apart. And you're going to actually do this in lab. You start off with leaves, spinach leaves, and the first thing you do is you throw your leaves into a Waring blender, just the kind that you have at home, not a very sophisticated scientific instrument. You blend it up and you get this green soup, and by differential centrifugation, which is shown in figure 38, put this suspension in a centrifuge and you start to centrifuge it at different speeds. The components of a eukaryotic cell are different sizes. So some will sediment very quickly, some will sediment less quickly, and some may not sediment at all. You can isolate fractions, and what's shown in figure 38 is that the Golgi apparatus would be at the top of this centrifuge tube, mitochondria would be at the bottom, and nuclei in the middle, and nuclei would be at the bottom. So you collect this one fraction, say you're interested in the nucleus, you collect the nuclear fraction, and you can then examine that fraction separate from all the other membrane-bound organelles that are present in the cell. Now, there's always criticism of this because you are looking at components and you're taking them out of their natural environment. They're not in there. You're not looking at an intact system anymore because you have smashed it apart. And you always have to be aware of that limitation when you use this sort of biochemical fractionation technique. So what we're going to be talking about today and Wednesday are eukaryotic cells. I'm not going to talk about prokaryotic cells very much. I may make reference to them every now and then. I think they will be discussed more thoroughly in the second part of the class. A eukaryotic cell has a nucleus, and then there's the region outside the nucleus. That region is known as the cytoplasm. And within the cytoplasm, there are more membrane-bound organelles. There are many of them, and multiple forms of them, I mean, numbers. So these are membrane-bound organelles. And what you can think of these as are little sacs with things inside them. And then there's another region. There's the region in which the membrane-bound organelles is floating. And that is called the cytosol. People often get the cytosol and the cytoplasm confused or mixed up. It doesn't matter. I think it's nice if we can use cytosol as this region that does not have the membrane-bound organelles and membrane-bound organelle. So this plus this equals that, the cytoplasm. Why do we have organelles? What's the general function of organelles? What you're going to see as I talk about specific organelles is each organelle has a specialized function. So you are taking a series of enzymes, proteins, and you're packaging them up into this guy here. This might be a mitochondrion. Mitochondria are energy-producing organelles, and this makes these processes much more efficient. You have the same processes occurring in prokaryotes, but in that case, you're more dependent on the fusion of molecules coming together because they're not localized in any specific region. So this complicated structure is contained in a localized region in a eukaryote. If you look at figures 39 and 40, they are simply to show you the complexity again of the membrane-bound organelles in eukaryotes. And there are a lot of different membranes inside this eukaryotic cell. They have very different functions, and they have how they work is very different. Some of the functions of the components in the eukaryotic cell are summarized in table 40, and you have to be aware of what each of these organelles does. I will talk about most of them. I think pretty much everything in table 40 I'm going to describe one way or another. So you have to be aware of what these things do. Now, one of the interesting features of, if you look at figure 39, one of the interesting features of these organelles within the cell is how they're made. It turns out that a typical eukaryotic cell, I've read this, I don't know if it's true, but I did read it somewhere. A typical eukaryotic cell may contain 1 billion proteins, 10 to the 10th proteins, 10 to the 9th proteins. Now, they're not all different proteins because there aren't that many different proteins. And I'll make a comment about that in a minute. So you have multiple copies of lots and lots of proteins. You have lots of different compartments in this cell. You have the circles, the squares, the boxes, the nucleus, the membranes, and one of the major important questions is what I've written on the board over there, protein targeting. Protein targeting. What does that mean? The question is, how do proteins get to the right organelle? Big, important problem in modern cell biology, which I will talk about because I think it's interesting, I think it's important. If I have a protein that's involved in the mitochondria, and for some reason that protein ends up in the lysosome, this is a dead cell, basically. Okay? So proteins have to end up in the right place. And I think at some point, fairly recently, people realized this. They said, well, my God, we're making a lot of proteins actually in the cytosol of the cell. We'll talk about this in more detail. And then they are directed to various places within the cell. How does that work? What's the mechanism? It can't be random. So that is one of the big deals of modern cell biology, and something that, as I said, we'll talk about. Okay, let's start off, and I've got enough time to talk about a couple of things. The plasma membrane. I will spend more time on membranes later in the week, I think. The plasma membrane is shown in figure 41, and I'm not sure I did this when I talked about, yeah, I did do this. Lipids. Remember that I said phospholipids spontaneously form bilayers, and I drew these things, these tadpoles, and I told you what they were. So you've got head groups, which are polar, and these side chains, which are nonpolar. And if you look at figure 41, you can see this is the basic structure of a biological membrane. You've got, for all intents and purposes in our discussion, you've got a phospholipid bilayer. But, as I mentioned, and I erased it, when I mentioned the plasma membrane cannot be impermeable to everything. It has to have mechanisms for allowing molecules in and out of the cell. So there are components that will cross the membrane, and they will cross the membrane, which again, remember, very hydrophobic. Let's talk about a sugar. How do you get a molecule that is very polar, like glucose, across this membrane? Well, there are proteins in the membranes. There are specific kinds of proteins, most of which are involved in transporting molecules across membranes. Very, very tricky process to transfer highly polar molecules across a very hydrophobic membrane. And there's a whole lecture on biological membranes. Some of the proteins that are found in the plasma membrane, as shown in figure 41, have carbohydrate groups. That's shown to the right, carbohydrate side chains, which are attached to those proteins. So some of these sugar molecules we've talked about are covalently bound to proteins. Those stick out of the cell. They're always on the outside. So you've got a carbohydrate, CH2O, carbohydrate group, sticking out. And they serve as markers. So there are, in other words, liver cells will have a certain group of carbohydrate molecules that say, I'm a liver cell. Nerve cells will have something else so that the right things come into the right place. Again, very, very important. So that's the plasma membrane, and I won't say more because we're going to talk about these proteins that are in membranes. They're interesting. They're not like the proteins I talked about last time. They have different properties. They have different structures. The nucleus. I have enough time to talk about the nucleus. That's generally the largest organelle in the eukaryotic cell, and you can see that in both figures 37B and C. It has an envelope around it. It has a membrane around it. That membrane has pores in it, and I'll talk about these pores on Wednesday, because there is trafficking of things in and out of the nucleus. Remember, the nucleus contains genetic information, and that information has to somehow be transferred out of the nucleus, out to the cytosol. Again, there's a very important word I want to say now. Almost all of the genetic information, all of the nucleic acid in a cell, a eukaryotic cell, is in the nucleus. And I emphasize almost all, which means not all. And it's interesting and important to discuss where else DNA and RNA are found and what is the origin of that DNA and RNA are found. The DNA is organized into these chromosomes, and at various times the chromosomes do this wonderful process of mitosis. They separate, they are duplicated, and you can end up with two different kinds of, two numbers. There is a region in the nucleus known as the nucleolus. And usually it can be seen as a darker region in electron micrographs of cells. And the nucleolus is a region which is involved in the synthesis of something I've talked about already, mentioned ribosome synthesis. What are ribosomes and why are they important? Ribosomes are particles, not organelles. We lived with Campbell's textbook for 8 editions where he called ribosomes organelles. And they're not organelles. They're not surrounded by a membrane. They are particles that contain RNA plus protein. And what they do is more important than, well, it's equally as important. They're basically the machines which make proteins. So ribosomes are involved in protein synthesis in cells, all cells. That's why I said, what's another property? You know, got outer membrane, genetic information, and I think ribosomes should be mentioned in that context. Wherever you have ribosomes, you have proteins being synthesized. If you don't have ribosomes, you don't have proteins being synthesized. And you will see there, turns out there are some organelles other than the nucleus that contain ribosomes, suggesting they are actively involved in protein synthesis. Ribosomes are made in the nucleus. Let me back off. Ribosomes, there are 2 different subunits. Large, they're just, and small. Isn't that interesting terminology? There are, an active ribosome has 2 different subunits, a large subunit and a small subunit. They combine when proteins are being made, and we'll go through this in more detail, but they're made in the nucleus and they are exported out of the nucleus. So here's an example where you have rather large, complicated structures, the large and small subunits of the ribosomes, which are being pushed through a membrane. These ribosomes being made in the nucleus, the RNA is in the nucleus already, but there are proteins that combine with the RNA to make the subunits. That comes in. So you've got protein going in, and you've got subunits coming out. Very bad figure. You can't see any of that. And the various subunits that are made combine in the cytosol of the cell to become active in protein synthesis. I'll stop there because it's a good stopping place. I'll go through this again on Wednesday because you have to understand this to get what's coming later.